Welcome everyone. Today we will explore the specialized type of cell division, that is meiosis. Meiosis is the basis of genetic diversity. A deep dive into the cell division for gamete formation. Friends, we have discussed the mitosis in last video. Today we are focusing on mainly meiosis, which is very much important and basis of genetic diversity. In this video, what we are going to learn? What is the purpose of meiosis, stages and their key events, how meiosis contributes to genetic variation, and comparison between mitosis and meiosis. What is meiosis then? Meiosis is a type of cell division that reduces the chromosome number by half, producing the 4 genetically unique gametes, which were not produced in mitosis. As a mitotic division, only the 2 daughter nuclei were formed, but in meiosis you are having 4 daughter nuclei which are genetically different. As we have discussed, purposes are there. Number 1 is to ensure the correct chromosome number in each and every generation. Number 2 introduces the genetic variation by means of sexual reproduction and where it actually occurs. Particularly, it occurs in germ cells of sexually reproducing organisms. Without meiosis, the genetic variations may not come or arise. Where meiosis occurs? In animals, it occurs in testes in males and ovaries in females. In case of plants, it occurs in anthers, that is male reproductive floral part, and in ovule is a female reproductive floral part. Whereas in fungi and protists, it occurs in the form of spore formation. In meiosis, 2 successive divisions takes place. Number 1 meiosis first and number 2 is meiosis second. What exactly happens in meiosis first? Meiosis first is also known as a reductional division, whereas meiosis second is known as a equational division. It means the reduction only takes place once, and in second meiosis only equation takes place. What are the phases of meiosis first then? The prophase first is subdivided into the 5 substages: leptotin, zygotine, pachytine, diplotin and dikinesis. We will see step by step each and every substages of prophase first. Prophase first is somewhat complex stage of meiosis. In leptotin, you can find the chromosomes as a loose ball, that is long thread like structure that are loosely interwoven. In zygotine, where the pairing of homologous chromosomes takes place and bivalence has 4 strands, and the third and most important event takes place in zygotine is 0.3% DNA replicates during this stage. Then what about 99.7? It gets replicated only in interphase. The third substage of prophase first is pachytine, where non-sister chromatids of homologous chromosomes may exchange these segments or regions of homology is known as crossing over. It means the actual crossing over takes place in pachytine and it is in between non-sister chromatids of homologous chromosomes. Diplotin, where the synaptonymal complex degrades and homologous chromosome separates from one another a little. The chromosome themselves uncoil a bit, allowing some transcription of the DNA. And the last stage of prophase first is dikinesis, where the chromosome condenses further during the dikinesis stage, and this is the first point in meiosis where the 4 parts of the tetrads are actually visible. After prophase first, the homologous chromosomes are arranged at the equatorial plate in the metaphase first, where you can see the color combinations of the chromosome. Those chromosomes are looking different, those are not similar. Why it is so? Because the crossing over took place earlier only in this Pachytene stage of prophase first. Because of that only the chromosomes are looking different. After metaphase first, the cell enters into the anaphase first, where the homologous chromosomes pulled to opposite poles. Here also you can see the color combination is different. It means the recombinant homologous chromosomes are pulling apart. But in case of mitosis, what exactly happens? The sister chromatids get separated in anaphase, whereas in meiosis first and anaphase first, the homologous chromosomes are being separated. After anaphase first, when cell enters into the telophase, the daughter nuclei are formed, which are 2 in number, but genetically haploid in nature. After telophase first, the second meiosis starts, that is, daughter cells undergo a second division much like mitosis. No additional replication occurs. Prophase second, where the spindle fibers form again to separate the sister chromatid. When we are talking about, it is just like mitosis, it means in meiosis second the sister chromatids will get separated, whereas in meiosis first homologous chromosomes get separated. That is the major difference in meiosis first and meiosis second. In metaphase second, again the chromosomes, individual chromosomes get aligned at the equatorial plate. In anaphase second, the centromere splits and sister chromatid starts to separate from one another, and it is being pulled with the help of spindle fiber. At the end of the telophase, the 4 haploid unique daughter cells are formed, which are originated from one diploid cell. At the last, the cytokinesis takes place. With the help of the cytokinesis, all 4 haploid daughter nuclei get separated. What is the significance of the meiosis? It maintains the chromosome number across the generations. If the meiosis does not take place, it means the chromosome number may get increased from generation to generations. If suppose that in human being 2n is equal to 46 is in one generation, if the meiosis is not there, then the chromosome number will get doubled in next generation. Introduces the genetic variation, which is essential for the evolution of new life on this earth planet. If any errors occurs in meiosis, it can lead to genetic disorders like Down syndrome, that is trisomy, where the entire chromosome number, that is at the position of 21, the additional chromosome number may get added. Because of that, the genetic disorders may arise. So friends, I hope you all understood what I just explained. If you actually learned, I would like to test your learning. You just go through the MCQs which I have mentioned here, answer those MCQs and test your knowledge. Students, keep learning, be in touch for the new videos